Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Biomedical and genetic engineers at Duke University and the Albert Einstein College of Medicine have designed a small fluorescent protein that emits and absorbs light that penetrates deep into biological tissue. Tailored to wavelengths in the near-infrared, NIR, spectrum, this protein can help researchers capture deeper, cleaner, more precise biomedical images. This work appeared December 1 in the journal Nature Methods. Imaging deep tissues with light is challenging. Visible light is often quickly absorbed and scattered by structures and molecules in the body, preventing researchers from seeing deeper than a millimeter within a tissue. If they do manage to probe further, substances like collagen or melanin often muddy the image, creating the equivalent of background noise through their natural fluorescence. Biological molecules naturally absorb and emit light in the visible spectrum, which is about 350 to 700 nanometers, said Junji Yao, assistant professor of biomedical engineering at Duke. So when using it to image deep tissue, it's like trying to observe the stars in daylight. The signals get flooded out. To wade out from these muddied waters, Yao and his collaborator Vladislav Verkisha, professor of genetics at Albert Einstein College of Medicine, New York, developed a protein that absorbs and emits longer wavelengths of light in the near-infrared, NIR, spectrum. Tissue is the most transparent in the 700-300 nanometer window of NIR light, said Yao. At those wavelengths, light can penetrate deeper into a tissue, and because there is less natural background fluorescence to filter out, we can take longer exposures and capture clearer images. Verkisha and his lab used a process called directed molecular evolution to engineer their proteins, US. These photoreceptors are useful for imaging research because they can switch between a silent and active state when hit with a specific wavelength of light. They can bind with biliverdin, a biomolecule that appears in high quantities in mammalian and human tissues. We studied the structure of the biliverdin to determine how the photoreceptor would best attach to the biomolecule, explained Verkisha. After we understood the binding process, we carefully introduced substitutions of key parts of the molecule connecting to the biliverdin to increase the electron binding, which is necessary to obtain the red-shifted fluorescence. Last, we performed random mutagenesis followed by high-throughput screenings so the proteins would evolve and increase their brightness. The brightest found protein, termed MIR-FP 718 nano, is easily produced in cells and tissues and emits light just outside of the visible range. But while the NIR activation itself is helpful, what happens after the initial burst of activity holds even more promise for biomedical imaging. We've seen that the NIR range can be broken up into two main zones, explained Yao. When NIR light first hit these proteins, they emit light in the first zone, which is about 700 to 900 nanometers. But as they decay, the wavelength gradually gets longer, like the tail behind a comet. That's when they begin to emit light in the second NIR zone, which is from 900 to 1300 nanometers. In this second zone, all benefits from using the shorter wavelength, Zone 1 NIR light are enhanced. Light can penetrate tissue 2 times deeper, background fluorescence is significantly dimmed, and IM. As a proof of concept, Yao and his team at Duke used an imaging technique called short wavelength infrared, SWIR, to test the efficacy of the new protein. This process sends near zone 1 light deep into the tissue to activate the fluorescent proteins. As the proteins decay, they emit near zone 2 light that provides information about the structure and composition of the targeted tissues and cells that can be translated into high-resolution images. After introducing engineered Mir-FP718 nanoproteins into their animal models, the team used them to capture images of microbes in the mouse digestive tract, visualize cells in a mouse mammary gland, and even track changes to inflammation in a mouse liver. All the images captured were clearer and more detailed than images made using a standard near zone 1 imaging protein. Yao and Verkisha are optimistic that their continued partnership will be a boon for their work in both biomedical imaging and protein engineering. As Verkisha continues to refine and improve the fluorescent proteins and biosensors, Yao is excited to use the new tool to visualize the brain more closely and to potentially track the movement of cancer cells. This is an exciting new front of our decade-long collaboration because we can use the imaging tools to guide the protein engineering decisions, and we can use the advanced protein engineering to improve the imaging capabilities, Yao said. This work was supported by grants from the U.S. National Institutes of Health, GM 122567, EB 028143, NS 111039, NS 115581, the Chan Zuckerberg Initiative, 226178, the Cancer Foundation Finland and Magnus Ehrnrooth Foundation, and the NIH Intramural Research Program for the Vaccine Research Center of the Materials provided by Duke University. Original written by Michaela Kane. Note: content may be edited for style and length. Olena S. Olenik, Chenchuo Ma, Sergei Pletnev, Mikhail Balaban, Carlos Toboada, Huaxin Sheng, Junji Yao, Vladislav V. Verkisha. Deep tissue SWIR imaging using rationally designed small red-shifted near-infrared fluorescent protein. Nature Methods, 2022, doi 10.1038, S41592, 022, 01683, O Duke University. Small glowing protein allows researchers to peer deeper into living tissues. Proteins that emit longer wavelengths of near-infrared light help create detailed, high-res biomedical images. Science Daily. Science Daily, 5 December 2022. ift.tt/piatextdc. Duke University. 2022, December 5. Small glowing protein allows researchers to peer deeper into living tissues. Proteins that emit longer wavelengths of near-infrared light help create detailed, high-res biomedical images. Science Daily. Retrieved December 5, 2022 from ift.tt/piatextdc. Duke University. Small glowing protein allows researchers to peer deeper into living tissues. Proteins that emit longer wavelengths of near-infrared light help create detailed, high-res biomedical images. Science Daily. https://ift.tt piatextdc, access December 5, 2022. Thanks for watching. Please subscribe my channel for more videos.